Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim.
Let us all praise Almighty Allah and the Prophet Muhammad, peace be upon him. Dear friends, it's well known that the world has faced the pandemic created by the COVID-19 virus with more than 2.5 million victims, 400 million infected, not counting economic impact. Due to the problem of the disease caused by COVID-19 virus and pressing epidemiological situation, researchers need to study and explore the process and mechanisms of development of viral disease. In the course of pandemic, there was no possibility to identify and study possible behavior of the COVID-19 virus in laboratory, the basis for clinical signs and symptoms of the viral disease. Presumed behavior of the COVID-19 virus is sustained by the empirical understanding of its structure. As it's well known, the COVID-19 virus consists of genome plus RNA, nucleoproteins, and surrounded by domain proteins E, M, and glycoprotein S, which serve as a receptor and so-called spikes. The COVID-19 virus is different from its coronavirus family, in that it is not surrounded by lipoprotein shells and therefore not able to preserve consistent morphology. Its morphology is in constant change in the form of single-cell amoeba. Such structural change in the COVID-19 virus were recorded 12,000 times by German virologists in 2020. Since COVID-19 virus lacks lipoprotein shells, it has low virulence for pathology. The virus virulence is represented by S-glycoproteins of spikes in the form of adhesive with bacteria. In the same epithelium organs there is a microflora. In the same epithelium organs there is a microflora in the form of supposedly pathogenic microorganism. This microflora is conditioned by symbiosis leaves of group of adenoviruses, coronaviruses, paragrips, and bacteria of Staphylococci, Streptococci family, and other bacteria. Among these, the family of Streptococci is different by its virulence. Of them, Streptococcus pneumococci is a diplococci. For most cases, with variability of virulence, the symbiosis life of COVID-19 plus Streptococcus pneumoniae happens in adhesive form. It's known that Streptococcus pneumoniae lives in microflora of airways, especially in the epithelium of the lungs, as bacterium attaches in the epithelium layer directly with F protein. In the layer of the epithelium of the respiratory path of the infected person, COVID-19 first interacts with conditionally pathogenic microflora, especially adhesiated by F proteins with Streptococcus pneumoniae. That is, the COVID-19 virus by S-glycoproteins receptors or spikes connect to F proteins of bacteria. Since symbiotic life of adhesive form of the COVID-19 virus with Streptococcus takes place in the epithelium layers of airways. In fact, the COVID-19 virus colonized Streptococci in the form of symbiosis. It should be noted that there is probability of colonizing other bacteria. It's known that Streptococcus through F proteins bind the layer of the epithelium. When the virus absorbs Streptococcus protein, it disconnects from the epithelium layer and ends up in mucous layer of the epithelium. Invasive factor causes production of hydrolysis of bacteria cells enzymes. The COVID-19 virus with Streptococcus in the wet layer of the mucous membrane epithelial epidermis is expressed to white-baked epidermocytes, Langerhans cells, which bind the virus and migrated through the lymph to the practic zones of regional lymph nodes, where it becomes antigenetic peptide T lymphocytes along with the class II HLA molecules. At the same time, it undergoes processing in lymphocytes. Symbiotic leaves of the virus Streptococcus bind, process, and overcome the obstacles with epithelial macrophages and the local dendritic cells and are presented to T lymphocytes. Once the barrier is overcome, symbiosis antigen passes to lymphatic vessels and then migrates further in the body. If an infected person has chronic disease in the airways, angina, tonsillitis, pharyngitis, bronchitis, and others, this place is a localization point from where the spreading of the symbiotic life of adhesive form of the COVID-19 virus plus streptococcus starts. It should be noted that the complexity and complications of the disease of an infected person depends on intensity of transition of the adhesive form of COVID-19 virus plus streptococcus to lymphatic fluid of the body. If in a low dose, disease is expected in light form, if in large quantities, in severe form. Migration of the symbiosis virus, as we have already noted, moves in lymphatic vessels inside the body. In this process, an infected person sweats a lot with no temperature and thirst. Since adhesive streptococci with the COVID-19 virus are reactive or pathological proteins, these can interact with proteins and blood cells receptors, can be absorbed by erythrocytes, neutrophils, bind to specific or semi-specific natural immunoglobulins, components of complement, binding proteins, monocytes, and blood lymphocytes. Proteins and cells binding the symbiosis virus are held in the main blood filter spleen, where it processed by macrophages. Lymphocytes are stimulated and cells producing antibodies are produced. The symbiosis virus is transported by blood through the body and binds with macrophages various organs and follicular dendritic cells in lymph nodes, Langerhans cells, killers, or the B cell zone of the spleen. Thus, white pulp spleen is targeted first, and at the same time, lymph nodes of the epithelium of airways, digestive, excretory system, and other internal organs. Liver is one of the most affected organs of the adhesive virus COVID-19 plus streptococci. It's known that in the spleen, B lymphocytes are produced. They are differentiated with white pulp, where immunoglobulins are synthesized. Symbiosis viral streptococci paralyzed immunoglobulins production, differentiation of T lymphocytes and B lymphocytes. This damaged the body depression and expression of antibodies of an infected person to some antigens, including chronic hepatitis A. Hepatitis A virus controlled by anti-IgG antibody comes out with adhesion of antibodies, actively multiply in geometric progression and expresses hepatocyte cells, normalizing composition of the blood and utilizing dead cells. These specific parts of pathology cause blood coagulation in the body. Blood coagulation leads to decreased mobility, activity, and expression of immune system. Thus, condition of person infected with COVID virus is complicated by an immunosuppressive state. The immunosuppressive state of patients weakens the primary protection of the immune system, primarily expressing T lymphocytes, B lymphocytes, and their derivatives of SD4, SD8, SD32, killers, local immunity, and others. At the same time, agranulocytes of immune cells, neutrophils, epicophils, basophils, and others face a decrease in activity in the result of blood thickening. Taking advantage of weakened immunity, symbiosis virus and streptococci, especially streptococci, produce pathogenic proteins that destroy the adhesive COVID-19 virus with streptococcal symbiosis in respiratory organs. It's clear to specialists that coagulation of the blood is initiated by the thrombosis of the arterial vessels of the body, especially in coronary and brain arteries, depression of immune system, structural destruction of layers of endothelial cells of blood vessels, including those caused by pooled arterial thromboembolia. Taking this opportunity, we should say a little about why thromboembolia occurs in pulmonary arteries and pulmonary pneumonia. As we said, coagulation of blood and immunosuppressive condition of the patient allows us to overcome pathogenic qualities of conditional pathogenical microorganisms living in the respiratory organs. First of all, we should recall a number of infections: pneumococcal, herpes, influenza, and parainfluenza viruses, adenoviruses, among bacteria streptococcal, staphylococcal, chlamydia, klebsiella, Escherichia, hemophilia, Mycoplasma. First of all, fungus in the form of candids and others. Since the pathological etiology of pneumonia depends on the species of the pathogens and its pathogenically. For example, influenza and parainfluenza viruses cause alveolar pneumonia near the bronchi and complication of chronic disease of the upper respiratory tracts. The hemophilic wound contributes to the development of pneumonia in people with chronic bronchial disease and sometimes smokers. Enterobacteria is a rare pathogen of pneumonia which affects mainly patients with renal failure and diabetes. Staphylococcus is rare pathogens of pneumonia but relatively often infect the elderly. Streptococcus is the most common cause by the development of pneumonia. Pneumonia of the lungs caused by streptococcus more often and fatally, in the case more than 80% damage of lungs and pulse oximeter reading of 65. Now let us see why thromboembolia happens in pulmonary arteries of COVID patients. Now those who follow closely the meaning of the article can see that adhesion of the virus Streptococcus forces Streptococcus to produce pathogenic proteins to get rid of the virus. These pathogenic proteins cause the destruction of a single layer endothelial cells and blockage of blood flowing in 3 layers of blood vessels: elastic, smooth, round, and connective tissue. Thus, the structurally changed vessels lead to coagulation of blood and separation of endothelial cells from the layer of blood vessels, causing thromboembolism. Thus, identified form of COVID-19 virus in no way damages the endothelial cells of the blood vessel, contributes to coagulation of blood, causes immunosuppressive state and other pathology of COVID patients, because it's subject to cell of immune system in all stages of disease of the patients.